Hello again. We're going to start a series of lectures on molecular biology now. Um, Before we do, I just ran across this quote a while back uh, from John Densmore about wanting to play music when he was in junior high school, and he had braces, and his dentist told him not a good idea to play the clarinet, and so he decided to become a drummer. And he became the drummer for a very well-known uh, rock band of the 60s called The Doors. So without his dentist, who knows what might have happened to The Doors. Anyway, um, I want to start talking about the organization of genomes um, and the relationship of those genomes between organisms. And we can, one way to look at this is to take uh, the genes, the same gene from different organisms, line them up and ask how similar they are to each other. And this is an example of three very divergent organisms, humans, um, the, an example of a different, from a different kingdom, the Archaebacteria, the bacteria E. coli, and then at the bottom, humans again, just to make it easier to compare. Now, where you see a little red dash, um, it indicates where the sequences are the same between the two organisms. And so you can see even when we compare, and this is a gene called the 16S gene that's present in all organisms and really has become the standard for comparing sequence information between organisms. But you can see that even between bacteria and humans, there's still a lot of similarity, or what we call homology. And so the closer two organisms are related to each other, the more homologous their DNA will be, or the more similar their sequences will be. Now we can use these kinds of information to construct these relationships between different organisms. And based on these sequences, we divide all of life into three different categories. On the right, under the blue band, are all eukaryotes. Those are complex organisms that contain a nuclei, basically, or subcellular organization. And you can see, although there's a lot of divergence, you can just see how in the big picture here, how close we are to other organisms, either plants or even single-celled organism like yeast. There's another kingdom that we're all familiar with over on the left, the bacteria, and then in the middle are the archaea, which probably aren't as popular. They are everywhere. We find them occasionally in the mouth. They're single-celled organisms, about as complex as bacteria, but really not any more closely related to bacteria than they are to eukaryotes. Now, in this figure, they've drawn it all back to what they're calling a common ancestor. I'm not sure that that's really a feasible endeavor to find the commonality between all these different organisms, but doesn't stop people from trying. Now, there are a number of processes that generate change in the genome, and some of those, the major ones are shown here. And of course, the first one is a change in the DNA sequence, or what's a single base change or mutation. And that's what we were looking at in the previous slide, are single base changes that had happened over a very long period of time. And so when we have organisms as diverse as humans and E. coli bacteria, there's quite a few differences. We also can get duplications. We kind of touched on that before in talking about resistant to drugs, and we can get, and we'll talk about it again, we can get resistant to drugs because they have acquired extra copies of the gene. Also, if there's a gene duplication, then over time those two sequences can diverge from each other and they can take on different activities. We can get this shuffling of segments of the gene, and again this can result in altered or new activities as well. And the final process here is horizontal transfer, where genes from one organism are acquired by another. And we see that quite readily in these antibiotic-resistant genes that move around from one bacteria to another, particularly if you have a lot of selection for it by using a large amount of antibiotics. The other thing we're seeing is in genetically modified plants that have been modified to be resistant to herbicide, and we're seeing that the weeds that we're trying to control are picking up these genes from the plants that have been modified, and they're becoming resistant as well. So these are 4 different ways that genomes change over time. And again, there can be combinations of them. We can get duplications, then we can get shuffling. They can then mutate, get single base changes over time to become different, and then acquire new activities or divergent activities. And this is an old diagram, but just showing some of what we call gene families. So what we have here are a number of gene families where there are multiple genes that are very similar to each other but have slightly or even not so slightly differences in activity. And so you can see there are some with as many as 38 to 77 members within that family. So these are presumably genes found in the genome that appear to all have been derived from the same progenitor, but now have different activities. And we see other ones that are in smaller groups, but almost half the genes that we know fall into one of these family relationships, and it's probably a much higher number now. This is from a textbook from several years ago. Now we see 2 types of homology between, or when we compare genes. On the left side here is an example of the genes diverging over time through reproduction. And so we have an ancestral organism has a copy of a specific gene. That organism reproduces, so now that gene is in both of the daughters or all of the progeny really, and over time then the progeny, the genes in those progeny can drift away from each other and take on slightly different activities. Now the other pathway on the right is what we mentioned a minute ago, we can have a duplication of a region of the chromosome that includes a gene. So now there are two copies, and now those two copies can diverge from each other. And let's look at a complex family of genes, in this case the globin genes that code for hemoglobin, or other globins, I guess. And so if you look at this, if you haven't looked at these kind of genetic trees, the vertical order does not confer any information. It's the horizontal difference between any two taxa here that are important. So you have to follow the lines out to a common node and then back to the other one you're comparing. So for instance, the fourth one down on the list, the chick myoglobin, in this diagram is right next to the one below it, the shark hemoglobin beta, although they're not very similar to each other. If you follow the lines out to a common place and then back in, you see that they really aren't very close, whereas the chick myoglobin is very closely related to the human myoglobin. So you can see now that there are many globin, or many genes in the globin family in humans, and some of them are close to other genes in the human genome. Some of them are close to genes in other organisms. So what we've had here is both duplication and divergence, I should say multiplication and divergence within a given organism, such as humans, as well as divergence over a long time, so that the similar genes in different organisms are not exactly the same. Now this is a picture of the human genome. There's 22 autosomes, the non-sex chromosomes, and then the X and the Y. And during meiosis or mitosis, where the chromosomes are fully condensed, so they're wrapped up as tightly as possible, certain stains give this this banding pattern, and this, unless there's been some weird rearrangement that has occurred, everybody's chromosomes will have this this banding pattern, and we can compare these to other organisms, or we can compare them to other examples in the same organism or other humans in this case. There are some rearrangements that cause some serious medical problems, whereas some rearrangements might not. The problem, if you have a rearrangement and involves two different chromosomes, so that part of one chromosome now is attached to another one, that could cause some problems in the next generation, because that generation, or that the genomes of the progeny may be either missing big chunks of DNA, or they may have a duplication of a large amount of DNA. Now we can compare the chromosome structure among different species, and what we see here, and this is a little bit complicated, and I'll go through it a little bit, but you can go through it in more detail, and you can see that we can take all these chromosomes from several primates and other mammals as well, and we can see that if through a series of rearrangements and inversions, we can make them all look exactly the same. So the different regions of the chromosome have been colored, different colors, and if we start on the left with what is considered the ancestral chromosome, and this would be chromosome 21 of humans, we see a single inversion now has put the blue that was started out at the end of the chromosome inside the yellow region in the pig. In the tree shrew, the chromosome's been broken into 4 different pieces. One of them attached, the red region attached to another chromosome, as well as the green looks like it's attached to a little chromosome. But basically, going from the ancestor to the shrew involved breaking this chromosome into 4 parts. I'll show you a pretty extreme version of that in a few minutes. The lemma involves just a fusion of the original chromosome to another piece of DNA, shown in white on that one, and so on. So you can see that you can have parts of this chromosome break off, parts of other chromosomes attached to it, chromosomes, parts of the chromosome being inverted within that chromosome. So all kinds of rearrangements take place over time. And once again, if you have severe rearrangements, it makes it very difficult to mate because that after meiosis and cell fusion, there'll be either a significant chunk of DNA lost or duplicated, and that usually leads to some severe medical issues, and usually that organism does not survive. Now, if we compare chromosome 1 from 3 primates here, again, we can see some similarities, a lot of similarities, particularly in the lower half of the chromosome, in the bottom half here, but also some differences. So this type of analysis, this banding analysis, has been very helpful in comparing genome structure. Of course, nowadays, when we have more and more organisms sequenced, this type of approach is becoming less powerful. Now I want to show you one more example, and that's 2 species of muntjac. One's called the Chinese and one's called the Indian muntjac. And although this picture tends to make them look a little different, I've been told by someone who actually studies muntjacs that if you put an Indian muntjac right next to a Chinese muntjac, you would be hard-pressed to tell the 2 apart. So they look almost identical, but they can't mate. They don't mate. They're not able to mate with each other. And this shows the reason why. If we look at the karyotype of these 2 mammals, we find that the Chinese muntjac has a large number of very small chromosomes, whereas the Indian muntjac has just a few very large chromosomes. Now, if we band these with the same dye, that's called Giemsa, that we were looking at in the previous slide, we can take the Chinese muntjac chromosomes and line them all up, and the banding pattern looks exactly like the Indian muntjac. But for some reason, they've been split apart into many little chromosomes. So you can imagine that it would be very difficult for these 2 organisms to mate. Now I want to show you an example of how quickly rearrangements can happen. And this is an example in bacteria. And what was done here was to compare the genomes of 2 different very, very closely related species. In fact, these 2 are the most closely related species to each other. The one species is called Tannerella forsythia. It's been implicated in periodontal disease, so it's an oral bacteria. The other species does not have a name because it hasn't been cultured and characterized, although we were able to sequence the genome from one cell of the species, and this is some work that was done by Dr. Bell, who will be talking next week, and that I've had a long-standing collaboration with. And so what we're looking at here is a comparison of the entire genome of these species. And one way to do that is to line up the DNA sequence of one genome on the x-axis and then the other genome on the y-axis, and then make a matrix, and everywhere those bases are the same, I put a dot in. And to make it a little cleaner, generally we're looking at set a threshold of like 5 out of 12 or 7 out of 12 bases in a row are the same, and then we put a dot in and we go every base along the genome. So when you compare two isolates of this uncultured species at the top, and you line them up where you get homology between the two, you get a line. And so what we're seeing there in the top figure is that the majority of this genome is in the same order and very similar to each other. Now, when we take either of those and we compare them to T. forsythia, again, this is its closest relative. We do not see any kind of similarity in the structure of these genomes. So even though these two species are very closely related to each other, the organism of the genome is totally different. We can't find really any similarity between them. So there have been so many rearrangements over a pretty short period of time that they no longer look at all like each other. Now, if we look, if we zoom in on the genome and look at just a small region, and we see in bacteria, the organization of the genome is to have genes very close to each other. They're usually fairly small. There's very few regions that don't code for anything. In this case, there's a small region between gene B and gene C that doesn't. We're looking over about 10,000 base pairs here. So that's out of about 4 million base pairs in the E. coli genome. The other thing you'll notice is that the genes, the coding region can be on either chromosome, so they can be transcribed in either direction along the chromosome. Now, in bacteria, genes that are usually involved in or gene products that are usually involved in a similar process are clustered on the genome. And this is one example, and these are called operons, and they're generally transcribed as one big RNA molecule, and then each coding region is translated separately. This is an example of called the tryptophan operon or trip operon, and all the enzymes involved in synthesizing tryptophan are present in this operon and are all transcribed together. This is a very efficient way of synthesizing all the enzymes that are needed for a certain process, because you don't have to worry about regulating them all separately. You just if you make one, you make them all. Now, in eukaryotes, things are quite a bit different. And if we start up at the top, the red line representing an entire chromosome containing about 3,000 genes, or 150 million base pairs, and we take just a little portion of that and blow it up into the second line, and we can see where the red bars are where genes are located. The white bars do not code for genes, so we can see, unlike in bacteria, genes in eukaryotes are spread out quite a bit. Now, if we take one of those genes and blow that up, what we see here are, again, this gene is spread out, where the blue bars indicate where the sequencing the sequence regions are for that gene, those regions that are going to be transcribed into RNA and then translated into DNA. The yellow region represents sequences in between the coding regions. Those are called intervening sequences or introns, while the coding regions are called exons. We also quite often see very large regulatory regions, and shown here, the regulatory elements are all upstream of the gene, and that's not necessarily the case in eukaryotes. We can have some of them downstream, we can have some in the middle of the gene. So now this gene is transcribed with both the introns, the intervening sequences, and the exons, the coding sequences, all transcribed as one unit. And then after transcription, the intervening sequences are removed by what's called RNA splicing to give a mature RNA that then can be used to translate into a protein. So this is another example, in this case a human chromosome 22, which is 48 million bases long. If we take a small segment, about 10% of that has about 40 genes, fairly tightly packed in this case. We blow up another region of that contains about 4 genes. Again, you can see the spaces between them. And then finally, looking at one gene, we again see the regulatory elements, the coding regions or exons, and the intervening sequences that are going to be spliced out after transcription in order to make a mature RNA. So in the mid-60s, it was shown that in eukaryotes, RNA is generally transcribed as in large pieces, so very long strands, and then it's as it moves out into the cytoplasm, where it's going to be translated, it becomes smaller, and it was never clear why that was. It was speculated that it might get trimmed off at the front or the back or both, or it might be, or RNA might be transcribed as several genes that are then chopped up into individual coding regions before they're sent out into the cytoplasm. And nobody thought that that there would be these regions inside the coding regions that needed to be removed or spliced out before it could be translated into a protein. And this is a depiction, a schematic of the first experiment that showed that there were intervening sequences that need to be spliced out. And what was done was to take cells infected with adenovirus, and adenovirus is a common laboratory model virus, but it also causes cold-like symptoms in humans. And in the late stages of the infection, pretty much all the RNA made in the cell is off the adenovirus genome, and it's almost all from one transcription unit. So there's only one really RNA being made at this time. So what was done was to isolate RNA in the late stage of infection when it was mostly, if not all, this one transcription unit, and to hybridize it back to the adenovirus genome. and then look in the electron microscope. And what was found was is depicted here in that the DNA, shown as the double red line, hybridized back to the RNA, but parts of it were looped out. So there were intermittent segments that were hybridized to the mature RNA, showing that there were these intervening sequences in the middle that needed to be removed before the RNA could be translated. Now, if we look at three genes, in this case, the gene we talked about last time, the dihydrofolate reductase gene, from three different mammals. I believe the one in the middle is the mouse gene, and then the other two, I believe, are the human and the hamster genes. What we see here with the dark purple bars depicting the exons or coding regions, and the light regions, the introns, is that the exons are pretty well conserved, look pretty much the same from species to species. There's a little bit of difference in the final exon, the sixth one, but that's really in the non-coding region. So it's extra DNA that's at the end of the gene. However, if we look at the introns, we see that they vary in size from one species to the next. And they not only vary in size, but they vary in content as well. So while the exons or coding regions are conserved, the introns can change much more rapidly. If you stretched out the DNA from each cell in a human, each human cell, it would be over 2 meters long. And so packed into that tiny space, we really need to have some organization. And this is how chromosomes are organized in all eukaryotics, basically. And they're wrapped around this group of histone proteins. And so histones form a protein ball, and the DNA is wrapped around each histone 2 times and then moves on to the next one, sometimes called the beads on a string. Those histones with the DNA wrapped on them are then further coiled up among themselves to package them even more tightly, and then those coils are coiled again until finally they're coiled into a pretty small space. And during mitosis, they're extremely tightly coiled, and that's the chromosome pictures. The stained chromosomes that we were looking at previously are the ones that are coiled up the most tightly. But DNA that needs to be transcribed can be looped out and free for transcription. And you can see on this picture a mitotic chromosome with the DNA looped out. So those areas of the chromosome would presumably be available for transcription if that was needed. Now, bacteria genes are very small. They don't contain intervening sequences, but eukaryote genes, human as well as including human genes, can be quite large. And this is just some examples of the variety and size of different human genes, starting at the top of the fairly small gene, the globin gene. Again, this is expressed in red blood cells, and it's a fairly small gene. It has 2 introns in it. Makes the entire gene about 1,500 bases. The mature RNA about 600 bases. We go down to the other extreme, and that's the dystrophin gene. That is over 2 million bases long, has the mature RNA is still 17,000 bases. So it's a huge RNA, and it has over 50 introns in it. So, so the number of introns and the size of those can vary from one gene to another. Now, in a highly transcribed gene, we can find these structures in the electron microscope. And the ribosomal RNA genes, and these are the genes that code for the RNA molecules that are part of the ribosome, are present in multiple copies in tandem arrays, so it's a little bit easier to find them. And what we see here is along those arrays, we see each gene is identified by the number of transcripts coming off those genes. So in the bottom picture, you can see a couple of them, one in particular on the right. And so transcription is starting at the left end of this gene, and it's going down the chromosome, and as it moves down the chromosome, the transcripts get bigger and bigger to give you what's often called this Christmas tree structure. Now, genome sizes can vary quite a bit, and this is a depiction of that, starting at the top with sizes of bacterial genomes, which are generally in the range of a few million bases. A few of them maybe even less than a million bases. Something like E. coli, about 4 million bases. Yeast is about tenfold more DNA, or the fungi do, starting with yeast, which is a little over 10 million bases in the genome. Plants vary quite widely, as you can see here, and some plants have an extraordinary amount of DNA. Insects are about 10-fold bigger than fungi, starting at about 10 to the 8th or 100 million bases. And if we skip down to mammals, we go up another 10-fold into the billions of bases. Once again, amphibians have a wide variety of genome sizes and can, again, be extremely large, whereas the newt is over 10 billion bases, or close to 30 billion bases. It's not because the newt has that much more coding region. All that DNA is non-coding, and it's not clear why it has so much extra DNA, but it has an extremely large genome. So I generally think of genome sizes going from bacteria with in the millions, fungi or yeast in the tens of millions, insects like Drosophila in the hundreds of millions, and then mammals like humans in the billions. So a tenfold increase when you go through all of those different types of organisms. This is a table showing that information again. So if you like to look at tables better than graphs, this will again show you sizes of typical genome sizes for different types of organisms. Now, one of the reasons that mammals have bigger genomes than insects or than yeast is because they have bigger genes. They have more introns in the genes, and this is showing you distribution of the number of introns in different genes in the yeast, Saccharomyces, and the fruit fly, Drosophila, and in typical mammals. And so you can just see that in yeast, most of the genes have just one exon, so no introns. And if they have introns, they have just a very few. Drosophila, most of the genes have introns, and they vary in sizes, and some um, large number of them, whereas mammals have even more introns on average and fewer genes that don't have introns. Now, the introns can vary in size as well. You see with the average somewhere between 500 and 1,000 bases, but they can also be very large, more than 100,000 bases. So the first complete genome was finished in 1995, so that was about 25 years ago now. And originally, the way people were approaching genome sequencing was to march down the chromosome one fragment at a time, and then use the end of that fragment to start sequencing the next fragment. And as you might imagine, this is a very laborious process. But a group, a private group, came up with another approach, and that was to break the genome into small pieces and sequence them randomly, and then assemble them all by overlapping them. So if they're broken into at random sites, these short fragments will overlap, and you can construct the entire genome using that approach. And so that's still the approach that's used, and it's the approach that's been used for all genomes that have been sequenced, including the human genome. The first draft of the human genome was published in 2001, and there were two groups working on it. A group at the Ventnor Institute, a private company, and then a large consortium sponsored by NIH that involves many different labs. Now, when you sequence a bacteria, it's pretty straightforward. It's only a couple million bases, and so you again break it into random fragments, sequence, and then put it all together by overlapping the fragments. In a genome like humans that contains a huge amount of repeated DNA that presents some additional issues. And so what was done is first to map the genome, so identifying specific markers along the genome, so that the sequences can be placed in relation to those markers. And like I said, there were two groups working on this. The NIH group did the mapping, and so the Ventnor group used that map as well. And so they both came up with a rough draft of the human genome at the same time. But this diagram shows how that was done. The stars show these sites, sequence tag sites. So these are markers along the genome. Then large fragments were made from the genomic DNA and put into E. coli. These are called BAC fragments, bacterial artificial chromosomes. And then those are broken into smaller pieces, and you can see at the bottom here, these little black lines represent these small fragments, so once you have enough of them, you can overlap them and come up with these consensus sequences, or called contigs. So in order to get an entire genome, you usually have to sequence several times the amount of DNA is there, and anywhere from probably 7 to 10-fold. But that's still faster than sequencing one fragment at a time and going down the chromosome piece by piece, especially nowadays when you can sequence a bacterial, well, I should say in a sequencing run that takes a day, you can sequence maybe 100 or more bacteria. So these are some of the first organisms that were completely sequenced, several bacteria, and since they're so small, you might imagine that they were much, and they still are, much easier to sequence, especially since they don't contain much repeated DNA. A few archaebacteria, and then kind of the typical model organisms in eukaryotes, Saccharomyces, a yeast. The Arabidopsis genome was sequenced. That's a small plant, and unlike many plants that have extremely large genomes, Arabidopsis has a small, very small genome. And just mentioned the Arabidopsis Stock Center that collects different strains of the plant, because this is a model plant used for studying plant genetics. The Stock Center is right across the street here in the biology department. So the nematode, C. elegans, was also sequenced, and that's a organism that's been used extensively to study developmental biology. The fruit fly, Drosophila melanogaster, and of course humans. And these were the first eukaryotes, but now there are many, many more organisms that have been sequenced. So when we look at sequence of bacteria, what we find, again, are a number of genes, and the two rings represent coding regions, and the one ring showing it on one strand and the other on the other strand. The origin of replication is shown in the upper left corner, and that's where DNA synthesis begins and terminates at the other end of the ring. And remember, bacteria generally have a single circular chromosome, whereas eukaryotes generally have multiple linear chromosomes. Now, early on, a number of oral bacteria were sequenced, and as well as the yeast Candida albicans, which is an oral pathogen. And so this is a list of some of the early ones. Third one on the list, Bacteroides forsythus, has changed names twice since this slide. Now it's called Tannerella forsythia, and we talked about it briefly a little while ago. But now several hundred oral bacteria have been sequenced, including those that have never been cultured. And we, as well as other labs, have been able to pull out single cells of these bacteria and sequence the genome from one cell. The first oral bacteria that was sequenced is this one, Porphyromonas gingivalis. It's pretty well established that it's involved in periodontal disease. It's been extensively studied bacteria, probably the most, that and strep mutans, the most studied oral bacteria. And again, this is a depiction of the genome, the two outer circles showing the coding regions. They're color-coded by function. I don't remember what colors mean, but the inner circles show other features of the genome, including P. gingivalis actually has a number of repeated sequences, insertional elements, and we'll talk about those later, but more than a lot of other bacteria. Again, that makes it a little hard to sequence for a couple of reasons. One is trying to sequence through them, and also placing them on the genome, because you can't sequence from one end to the other, so you don't know which end of the repeat goes to which other end of the repeated sequence. Now we can take that sequence information and construct a metabolic map. And this is shown for P. gingivalis and shows some of the enzyme activities that the bacteria has. Keep in mind, though, that every time we sequence a bacteria, we find genes, active genes, that we have no idea what they're coding for. So still a good fraction of the genome has not been assigned any function. And so every time we sequence another bacteria, we find new genes, and we also find genes we've seen before but still don't know what they code for. Now let's take a look at the human genome, and this is a schematic of what's present. So this colored line represents 100% of the genome. This was produced quite a while back, so probably a greater percentage of it has now been sequenced, but at the time, maybe 8% of the genome had yet to be sequenced, and most likely because it's full of repeated sequences that are difficult to sequence through. So if we start over at the left-hand side of the line, we see about 20% of the genome is made up of what are called lines, and these are short and interspersed nuclear elements. Okay, so these are repeated sequence. Sorry, these are long. L stands for long, as opposed to the signs next to it, which are short repeated elements. Okay, so a third of the genome is made up of these long and short repeated elements. Several percentage more are made up of retroviral-like elements. So these are remnants of viruses that have integrated into the genome. We have then a few percent of, again, what are called transposons, and we'll talk about those. Those are pieces of DNA that can move around in the genome. A few percent are made up of duplicated regions, and we talked about that a little bit already, that you can have extra copies of a particular gene show up in the genome, and so at any given time about 3% of the genome is made up of this duplicated regions. And you can, if you have a way of selecting for those, you can select those out, or select out cells that contain a particular region that's been duplicated. Then we have even more repeated sequences. And then we finally get down to unique DNA. And the pink area shows the introns, and the red area shows the coding region. So you can see that very little of the genome actually is used for coding for proteins. Introns make up much greater fraction of the genome, but also keep in mind that even though on this diagram the introns have been included in the unique DNA, there can be repeated sequences within the introns as well. Okay, so this is a pie chart showing kind of the same thing. Again, this little wedge up at the top is counts for the coding regions, just 1 or 2% of the genome. Introns and regulatory sequences account for close to 1/4 of the genome. There's some somewhat unique DNA that's non-coding, and then the rest of the genome is made up of these repeated elements. Okay, this illustrates actually the point that we still don't know what a lot of these genes are doing, and so we can divide the genes into by function and show on this chart, and this big region to the bottom right depicts the genes that we have yet to determine their function, and so makes up over a third, between a third and a half of all the genes in the genome. We still don't know what they're doing. You can look at the other categories here and just kind of see how much there is of each one of these types of proteins. So this is where things stand today, and well, obviously when you have billions of bases in the genome, you can't characterize those by hand. You need some sort of computer program to assemble all the sequence data. And so this is comparing 4 different assemblers, and you see they are giving very similar answers in most cases, but you can look through here and see where they differ. But basically, there appears to be about 20,000 coding genes in the human genome. And since many genes can code for multiple transcripts, there are anywhere from 150,000 to over 300,000 transcripts. And that probably depends on your definition of what's a unique transcript. If we look in this next table, this is a chromosome by chromosome distribution of those. I'm not going to go through these, just kind of for your information. Of course, when we number the chromosome, we start with the largest one and go down to the smallest. So you'll see that the size of the chromosome decreases as you go down the list. Okay, so one of the first mammals to be sequenced after the human sequencing was the chimpanzee. And so that's having the chimpanzee sequence then has allowed us to compare the genomes between humans and a very close relative and see what's the same and what's different. And so I want to take a look at that, but before I do, I just wanted to share this picture that I found several years ago in the journal Science, and it was a response actually to an article that I did not see the original article, but it appeared that in that original article they were trying to make the case that humans were the only species that use tools, and this picture was sent in as to refute that idea, and saying that in this case chimpanzees use tools as well. And well, we've seen other animals, birds use tools, but in this case what's going on here is the chimpanzee lying on the ground is having his teeth cleaned by the chimpanzee in the middle with a stick. So chimpanzees are using tools to do dentistry. I always think this is particularly appropriate for dental students, since this guy on the left here is going to be looking over your shoulder for the next 4 years, just like the one in the picture. Okay, if we look at the evolutionary tree that compares 4 different mammals, and again the horizontal lines indicate how different each species is from each other, and you can see that the rodents are quite a bit distant from the primates. Within the rodents, the mouse and the rat are somewhat distant from from each other, whereas humans and chimpanzees are very closely related at the molecular level. And so if we look across the genome at every chromosome, we see that for all the autosomes, the amount of divergence is fairly similar, around between 1 and 1.5%. And it's not clear to me if this is just looking at coding regions or total DNA. It's most likely looking at coding regions or unique DNA. The interesting thing is that the X chromosome is more conserved between humans and chimpanzees than the autosome, and the Y chromosome is less conserved. It's not clear why that would be the case. Now, if we look at just chromosome 1, again, this is very similar between the two organisms. We see in the top graph the amount of diversity across the chromosome, and we see a slight trend in the increase in diversity as we move out towards the ends of the chromosome. And again, looking at the red lines down at the bottom, the amount of recombination that increases as we move out to the edges of the chromosome. Now we can compare deletions and insertions of the two genomes, and we call those indels because when you compare two sequences and one has something that the other one doesn't, we never know whether the one organism has acquired additional DNA or the other organism has lost some. Okay, so you can't tell just by looking at comparing the two sequences. So what's shown here is a distribution of the size of these indels across the chimpanzee and human genomes, and you see a spike, around a little over 300 bases, where that seems to be a common size for insertion or deletion event. Now there are a number of these repeated transposable element-like sequences in both humans and chimps. And they're similar in both cases, although they vary in quantity. And here's a list of some very common repeated elements. And you can see, for instance, both organisms have quite a bit of what is called an ALU sequence, although humans have over twice as much, 2 to 3 times as much. ALU is an interesting repeated sequence because it was identified very early on, long before the genome was sequenced, because there are these short repeats. I believe there are just over 100 bases, and they all contain a restriction site for the enzyme ALU1. So each repeat is cut one time by this restriction site, making all these exact fragments of exactly the same size. And then when you run it on a gel in electrophoresis, because there's so many of these, they show up as a very strong band. And so when people cut the human genome with the enzyme ALU1, ran that on a gel, saw this very prominent band, and they called it the ALU band. Okay, again, the number of lines, these long interspersed nuclear elements, are very similar in chimps and humans, whereas some of the other ones are present in both, but at different amounts. Now, this table shows that different sequences, particularly different genes, are diverging at different rates. So when we compare different genes, between the human and the chimpanzee, we find that some of them are highly conserved, whereas other ones are diverging much more quickly. And this is always the case when we compare two sequences, and particularly, there are some genes that are under a lot of constraints in terms of any kind of variant will destroy the gene, and the histones are a good example of that, where when we compare the histones, remember, are the proteins that DNA wraps around in the nucleus to form nucleosomes. And if we compare the sequence of the histones between humans and the green pea, we find that there's only 2 or 3 different amino acids. Now remember, there's some redundancy in the genetic code, so we do find much greater difference at the DNA level, but at the protein level, they're virtually the same. Other genes may diverge very rapidly. One that I've looked at is a gene in the bacteria Porphyromonas gingivalis that codes for a protein on the outside of the cell, and it's a protein that's a target for host antibodies. So it is changing very rapidly to the point where you really, in many cases, don't see any similarity between different strains of P. gingivalis. Okay, so some genes, and mainly because of constraints, because they have to maintain their activity, are changing very slowly. Others are changing very rapidly. So I want to move on now and talk about DNA replication. Well, let's look at the next slide. And again, this is the idea of the central dogma of molecular biology, where you have DNA coding for a gene or genes. Those genes are transcribed into RNA, and then the RNA is used to translate that into a protein. And so for the next couple of segments, we're going to talk about what happens actually before transcription, or what's above, in this line, the red lines depicting the DNA. So we'll talk about DNA replication, DNA repair, because there are always insults to the DNA, and oftentimes those can be repaired back to the way they were, and also about genetic recombination, or the shuffling of the DNA on the chromosomes. So the first thing we want to talk about with replication is this idea of semi-conservative replication, and this is demonstrated in the picture on the right-hand side here, where we have a double-stranded DNA molecule, so we can separate those two strands and use each one as a template to synthesize a new complementary strand. And then now we've got 2 double-stranded molecules, and in the next round of replication, again, we can separate the strands, use them as templates to synthesize 4 new strands, and so on every round of replication. And that way we can preserve the sequence of the genome from generation to generation. Now the basic building blocks here are shown, and the basic reactions, where we have, again, the nucleotides, the sugar, phosphate, and the base come together to make the nucleotide. We talked about that last time. and then those are inserted into the developing strand opposite of their complement. So you can see in part C, we're inserting one base at a time, following these base pairing rules of G, C, and AT. So if there's a C on the template, G will be inserted in its place. And there are a number of polymerases in the cell that carry out different functions, and the next table is a list of some of them. And we characterize them by what the template is. So these are all template-driven polymerases, and so we characterize them by what the template is and also what they're synthesizing. So DNA polymerase is a polymerase that uses a DNA template to synthesize new DNA, and that's involved in replication. We'll also see later on that there are polymerases, DNA polymerases, that are involved in repair. RNA polymerase, as the name implies, still uses a DNA template, but synthesizes RNA, and so that's used for transcription. Reverse transcriptase is the opposite. It uses an RNA template to synthesize new DNA, and we find that in retroviruses, and we also see it in other cells as well. And the last one on the list, primase, is an RNA polymerase that uses DNA as a template that's also involved in replication, and we'll see how that works in just a minute. Now, one important thing to remember is that all known polymerases, whether they're RNA or DNA polymerase, synthesize the RNA or DNA in the 5' to 3' direction. So there's directionality here, and we'll look at that in a second. But they cannot go backwards. They only go in one direction. And that direction is defined by the pentose, the sugar part of the molecule. And we label the carbons 1 to 5, and to distinguish them from the carbons and nitrogens in the bases, we add a prime, so 1' to 5' for the sugar, carbons on the sugar. So when we look at those positions, the only one that really we don't need to talk much about is the 4'. The 1' carbon, as we saw last time, is where the base is attached. Remember, the synthesizing a purine, it starts on the 1' carbon and is built from there. The pyrimidines, you make the base first and then attach it to the 1' carbon. The 2' carbon is where we see a difference between RNA and DNA, and that's shown on the right-hand side here, where there's a hydroxyl group at the 2' carbon on ribose, or an RNA, and a hydrogen, or and deoxyribose, or DNA. Now the 5' and the 3' carbon is where adjacent nucleotides are attached, and that's what confers this directionality. Okay, so as we build a chain, we're going to attach the new base to the 3' carbon of the nascent DNA chain and build it going from the 5' to the 3' direction. And that's shown here, the short length of DNA showing again at the top the 5', at the bottom the 3', and these bases are linked together through this phosphate that forms phosphodiester bonds. between the phosphate and the sugars. And so as we're synthesizing a new DNA chain, the bases will be attached one at a time to the 3' end here at the bottom. It shows it a little differently. Again, 5' at the top, and the new bases will be attached at the bottom at the 3' hydroxyl group of the existing chain, the last base on the chain. And once again, the template will be used, or the old strand of DNA will be used as a template following these base pair rules. So if there's an A on the template, a T will be placed in the nascent chain. If there's a T on the template, an A will be. Likewise with C's and G's. So A's and T's always pair, G's and C's. Now one thing to notice about the base pairing is, and we probably will be coming back to this, but the base pairing between A's and T's involves hydrogen bonding between in 2 locations between the A and the T, whereas in a CG base pair there's 3 hydrogen bonds formed. And that makes the CG base pairing tighter than the ATs. And so if you have a run of DNA with a lot of A's and T's in it, it's going to be easier to separate those strands than if you have a lot of G's and C's. And one way to separate the strands in the lab is to just heat them up until you melt them apart. And if it's a highly GC rich sequence, you have to heat it to a higher temperature. Okay, so this shows the reaction then. We have our nascent chain over here on the right, upper right. We're going to add a base to it, and that's added as the triphosphate, shown in the gray square here. And as it's added, the pyrophosphate, or 2 phosphates, are cleaved off. And so then the new base the new nucleotide attaches to the 3' hydroxyl group on the nascent, the N base on the nascent DNA molecule. So that proximal phosphate on the incoming nucleotide, shown in the blue circle, you can see is added down here at the bottom to the chain, whereas the other 2 leave as diphosphate or pyrophosphate. We call that the alpha phosphate as opposed to the gamma phosphate at the end of the 3rd phosphate out, the distal phosphate. Now again, we're talking about template-directed synthesis, and these polymerases are template-directed, all the ones I listed on that previous slide. There are some non-template-directed polymerases. I think the one that comes to mind is an RNA polymerase, poly-A polymerase, that attaches strings of A's to the end of messenger RNA molecules in eukaryotic cells. But here we're talking about template-directed synthesis. So again, we have our old strand of DNA, and we're going to synthesize a new strand using that old strand as a template and putting in the complementary base as we go along, one base at a time. Now notice the other thing, that these strands are in opposite orientation. So the nascent chain shown here has the 5' end at the top and the 3' at the bottom, whereas the template strand has the 3' end at the top and the 5' end at the bottom. So that's the directionality. Again, this shows a template strand on the bottom here, a nascent DNA strand, a triphosphate coming in, cleaving off the pyrophosphate and inserting that nucleotide with one phosphate into onto the end of the nascent chain. Now, one thing about DNA polymerase is it's an elongating enzyme. In other words, it can attach new bases to the end of a nascent chain using a template to direct that, but it can't initiate the synthesis. So it needs something to synthesize off of or to elongate off of. And that's where this polymerase, the fourth one on the list, shown in blue here, RNA primase, comes in. So what is done during replication, since DNA polymerase can't initiate the process, is to synthesize a small RNA primer using primase, and then elongate off of that primer. Then to come back later and remove the RNA and fill in the DNA with elongating off the DNA fragment that was upstream of that. We'll look at that in a little more detail. But I want to get back to the DNA polymerase because in addition to its elongation activity, its ability to put in new bases along the, to elongate the nascent chain, the replicating DNA polymerase has what's called exonuclease activity, which means it can chew off or degrade DNA from the end. There are two types of nucleases, endonucleases that cut in the middle of DNA and exonucleases that chew off a base at a time from the ends. Now there are two types of exonucleases, ones that chew from the 3' to the 5' direction, that would be backwards from the direction of synthesis, but there are other nucleases that chew from the 5' to the 3' direction, and we'll get to those sometime later. So the polymerase uses this exonuclease in what's called proofreading activity. So after it puts in a base, then it looks to see if that base is the right one. Is it complementary to the template strand? And if it's not, then it can use the exonuclease activity to chew that base out and then put another one in and try again, try to get the right one. So again, if we have a base that's put in the wrong one, the polymerase can remove it if it's a mismatch, and then it can try again. Now in E. coli, and this is true for most organisms, the proofreading reduces errors in the polymerase by about 100-fold. So the error rate without the proofreading activity is about 1 in 2 million bases. So, you know, in this case, every time the E. coli replicated, there would be on average 2 errors. Now, with the proofreading activity, the error rate is reduced to 1 in 200 million bases. So every time it replicates, it probably doesn't make any mistakes for the most part. So it can replicate several times without making any mistakes, and that's a pretty good rate of fidelity. You know, if we were able to limit our mistakes to 1 in 2 times 10 to the 8th. That would be pretty nice. However, you can imagine if the human DNA polymerase makes a mistake in 1 in every 200 million, that there are going to be several mistakes made, several wrong bases put in every time a human cell replicates. Now, some of those might be corrected after polymerization, but other ones are carried along. Now, the fact that only a very small percentage, 1 to 2% of the genome is coding for anything means that even though there are some changes made every time the cell replicates, most of them are not going to be in coding regions either. So every round of cell division, there are probably a few mistakes. Most of those get corrected. Those that are not corrected are more likely than not to be in a coding region. Okay, just to summarize the different functions of polymerases, of nucleic acid polymerases. Again, DNA polymerase is a DNA template, DNA synthesizing enzyme. It has elongation activity, cannot initiate synthesis, but it also has exonuclease activity that can be used to chew out DNA if it's not the right bases. RNA polymerase can initiate synthesis. Again, this is an RNA-directed or a DNA-directed RNA polymerase, and it can elongate as well. Reverse transcriptase, remember, is a RNA-directed DNA polymerase, so it uses RNA as a template to make new DNA. Again, like other DNA polymerases, it can't initiate synthesis. It can only elongate. And finally, primase, which is used to make these primers so that DNA polymerase can elongate off them, is a DNA-directed RNA polymerase. And it can initiate as well as elongate. So now if we zoom out and take a look at what's happening where replication is taking place, this diagram illustrates that. And what we have here is a replication bubble. So there's an origin of replication in the middle of the bubble. And so on the extremes here are unreplicated DNA. In the middle is replicated DNA, so there's 2 double strands, the double strand being represented by the gray line. So DNA synthesis starts at this origin, and it moves in both directions away from the origin to make this bubble. And so as you continue to make more DNA, the bubble gets larger, and the unreplicated DNA gets smaller. Now, as you recall, all polymerases, whether they're DNA or RNA polymerases, can only synthesize nucleic acids in the 5' to 3' direction. So when we look at the replication fork in this diagram, and that's where the fork is where there's unreplicated DNA on one side and replicated DNA on the other side. And one of the strands is being replicated towards the fork and one away from the fork. So we call those the leading and lagging strands. And so the leading strand is synthesized continuously, but the lagging strand has to be done in short pieces because it's going away from the fork. So as the fork opens up, the polymerase can come back in and synthesize a new piece of DNA, which is often called an Okazaki fragment, named after the person who first identified them. So again, the leading strand is synthesized continuously, the lagging strand is synthesized discontinuously. So the polymerases are actually moving in opposite direction at the replication fork. Even though the general direction is moving in the same direction for both the leading and lagging strands. In the case shown here, the fork is migrating from left to right, like even though the top strand is being synthesized from right to left. Now, again, the polymerase, DNA polymerase cannot initiate synthesis, so it has to have something to elongate off of. So every time we start a new Okazaki fragment, the primase has to come in, make a short RNA primer, shown in black here, and then that's used to elongate off of with the DNA polymerase. So now you have a mosaic of DNA and RNA, and so RNA ribonuclease has to come in, remove the RNA primer, chew it out, and then the DNA polymerase can come in and fill in the gap. And then there's another enzyme that links them all together called a ligase. And I'll show you what happens if you have a defect in the ligase later on. Okay, so again, the lagging strand synthesis is in the opposite direction of the migrating replication fork, so it has to be done in small Okazaki fragments. Each one has to start with an RNA primer. Once those fragments are synthesized, ribonuclease has to come in, chew out the RNA, and then a DNA polymerase can come in and fill in the gap, and then DNA ligase can hook all these short fragments together. Now, and maybe we'll get to that later, but all this is done in one big conglomeration of proteins that has the two polymerases. And in eukaryotes, the polymerases on the leading and lagging strand are different. In prokaryotes, it's the same polymerase. Along with a lot of the enzymes that are needed. We've talked about some when we talked about nucleotide metabolism that are going along with the polymerases to make the deoxynucleotides that will be incorporated into the new DNA strands. So this is another depiction of the synthesis of Okazaki fragments. In the top figure, we see again the template as a red bar going from the 5' to 3' direction. So the synthesis on the line above that is going in the opposite direction from right to left. And so again, this would be moving, synthesizing away from the replication fork, so it has to be done in small fragments. And so we start with an RNA primer shown here as a little blue bar, and then that is used to elongate off of with the polymerase, in this case DNA polymerase alpha. Then the polymerase has to jump off and move up the chromosome towards the replication fork. The primase has to come in, make a new primer, and then the polymerase can elongate off of that one to make another strand, and then we're left with a series of small fragments that all start with a few bases of RNA. which then needs to be removed by a ribonuclease so that a DNA polymerase can come back in, fill in the gap, and then DNA ligase can hook all those fragments together. Now, when we look at the initiation of replication here starting at a specific location called the origin of replication, and as we'll see in bacteria, bacterial chromosomes have just one single origin of replication, whereas eukaryotic genomes have many, many origins. So this is showing one origin of replication where the two strands are separated, and then synthesis starts on both strands going in opposite directions to make this replication bubble. Now remember, on one replication fork, the leading strand is going to be on the top strand. At the other replication fork, it'll be on the bottom strand because of the leading strand heading into the replication fork, whereas the lagging strand, again, the synthesis is away from the fork. Now, if we look at the replication fork in more detail, there are a number of proteins here that are carrying out different functions. The yellow ball right at the replication fork it represents DNA helicase. The function of the helicase is to separate the two DNA strands so that the polymerase can get in and use them as templates to synthesize a new complementary strand. Now, if there was nothing else there, the helicase could go down the chromosome separating the two strands, but they would just come right back together right behind it. So there are these single-stranded binding proteins shown as little, looks like little footballs here, that hold the two strands apart after the helicase separates them. And this allows the polymerases to come in. Now, this is depicting a eukaryotic replication fork, as we see that on the leading strand, the DNA, or the polymerase that's synthesizing the DNA is DNA polymerase delta. On the lagging strand, it's DNA polymerase alpha. In prokaryotes, there's only one polymerase that does both the leading and lagging strand, and we call it DNA polymerase 3. So the first polymerases were isolated and characterized in bacteria E. coli, and they were numbered so that in the eukaryotic polymerases we use the Greek letters. And as I just mentioned, it's polymerase 3 in E. coli that is the replicating enzyme. And the reason for that is when the first polymerase was originally isolated and shown to be a DNA polymerase, it was thought at first to be the replicating enzyme, but it was quickly learned that it was not. And it was actually used to repair DNA. So a second one was isolated and called DNA polymerase 2, and it again thought to be the replicating enzyme, and again it was a repair enzyme, and it was polymerase 3 that was actually the replicating DNA polymerase. Now the reason that was the third one that was identified is pretty reasonable because there are hundreds of thousands of copies of Pol1 in bacterial cells where there are only just a handful of copies of Pol3. So lots of copies of the repair enzymes and very few copies of the replicating enzymes. Okay, back to the eukaryotic fork now. So we have the helicase opening up to two strands, the single-stranded binding proteins holding them apart, and then the polymerase delta on the leading strand heading towards the replication fork, whereas the polymerase alpha with its associated DNA primase heading away from the fork. Now this picture shows the primase interacting with the helicase, so it's backed up on the lagging strand to initiate a new Okazaki fragment by making a short primer. Then the polymerase alpha can then go back and start elongating off of that primer. So the steps required for priming, again, starting with the helicase, which I should point out requires ATP in order to separate the strands apart. And you might think that's reasonable that it would take some energy in order to pull the strands apart. Again, if they're highly GC rich, it's harder to pull them apart if it's because there are 3 base pairs per, or 3 hydrogen bonds per base pair, whereas if it's AT rich, it's a little easier to open up the strands. And so a lot of times we see more AT rich regions where replication starts. Okay, so after the strands are separated with helicase, the single-stranded binding proteins bind to it. Then that allows the primase to come in and start the primer, as shown in this picture. And once there's a primer, then our DNA polymerase can elongate off of that to start making DNA. Now the next picture shows a schematic of the single-stranded binding protein, and you can see what's going on here is that the proteins are sequestering the bases, so they can't base pair with the opposite strand. And of course, it can't be that tight of interaction, or it would be difficult for them to come off. And so the primase and polymerase have to push them off the proteins as they move down the strand and make a new DNA chain. Now there's one other molecule, or actually 2 here, that we want to talk about, and that's this sliding clamp. And it's this ring-like protein that goes around the DNA and holds the polymerase on the template. There's also a protein that facilitates that, the clamp loader. Now, all polymerases, DNA and RNA polymerases, have what we call a processivity. In other words, how long they will stay on the template strand before they fall off. And some polymerases have a high level of processivity, some have a less, are less able to stay on the RNA or on the template for very long. Some can stay on for tens of thousands of bases, but the clamp holds them on and allows the polymerase to go on for longer distances before they fall off. Now, if they fall off, of course, they can come back on and start synthesizing again. Okay, so when we look at eukaryotic chromosome, there are some important features in addition to the coding regions. These are shown here. Of course, the origin of replication, and again, in a eukaryotic chromosome, there are many origins. Otherwise, it would take forever to replicate something like the human genome. There's a centromere, which is responsible for pulling the 2 homologs apart after they've been replicated. And there's specific features on the ends of chromosome called telomeres, which we'll talk about shortly.